Hello children. Now today we will see something about crossing over and sex determination. See children, in case of crossing over, this we have already studied in our previous classes. Okay. Crossing over, what we can see, I think so this we have read in class 11th also, as well as some points we have seen in class 10th also. That is, during the crossing over, like we can say that at the time of meiosis, meiosis can be of two types, meiosis 1 and meiosis 2. Then we can see in case of meiosis 1 there is prophase 1, and prophase 1 can be divided into various parts. Okay, that is during the synaptonemal complex formation. There are the cases zygote and diplote, pachytene. This you have already studied. So what happens during the zygote and pachytene? The chromosome numbers, that these chromosomes, they make their copy, and after that these chromosomes mixed together and then they interchange or exchange their material. This is called as the crossing over. See, mutual exchange of, what we can write down, it is the mutual exchange of what? Blocks of homologous gene. Okay, homologous gene, and these genes are present from the homologous chromosome. What we can say that if this is a chromosome and suppose the genes are present here, then the mutual exchange of the genetic composition, exchange of this genetic composition, it will go to this side and it will come to this side. Mutual exchange is called as the crossing over, and basically this crossing over occurs in the meiosis 1 during pachytene and zygote stage. Now, how it occurs? See, these chromosomes make their copy. That is because of the replication, like this. Okay. Now, the copies, or I can draw it like this. This is the one more copy. Okay. Now what happens? Suppose if I draw one more. Now see what happens. This homologous pair of chromosomes, they will come close together. Okay, they will arrange themselves in this way, like this, and the red one will come here. And during this arrangement, there between these non-sister chromatids, there will be the exchange. Now, what type of exchange we can see? This red portion may come here and this black portion may go there. Okay. So, this is called, now how it has happened? It took place because of this structure. This will move to this side and the red one will come and cross like this. Okay. Now, this is called as, this point is called as chiasmata. The spelling is, okay, first crossing over. Now, this is all about crossing over. Because of this, now what type of structure we can get? We can get the structure like this. Understand? So this is called as the crossing over. Okay. Now because of the crossing over, the genetic material will get exchanged. This we have already seen in our previous class also, where I was teaching you the linkage, that the genes, they do not assort independently. Even during the crossing over, if the chromosomes they are shifting, they are moving, then the genes present on them, they can assort themselves independently. Okay, so this is all about the crossing over, children. Now the next is sex determination. Okay, autosomes and sex chromosomes, that is sex determination. Now see what happens. In 1903, there was a scientist. Okay, let me. Topic is sex determination. Okay. In 1891, one scientist was there and his name was Henkin. Okay. Now, what Henkin was doing, he was studying the process of spermatogenesis. And we know the meaning of spermatogenesis, that is the method of formation of sperms. So, Henkin was studying on the spermatogenesis. He was simply studying each and every structure of the spermatogenesis. And while studying that, he surprisingly saw the nuclear structure and found that during the process of spermatogenesis, inside the sperm, there are some extra structure. Okay, that is, he saw that the 50% of sperms have extra body and 50% of sperm doesn't have that extra body. Okay, understood. See what I mean to say, that while observing the process of spermatogenesis, Henkin saw that 50% of the sperms, they have one extra body and the 50% they did not receive any extra body. Now, Henkin said that as an X body, because it was extra body, so he gave the name as an X body. Later on, after many discoveries and inventions, other scientists, they came forward and then they said that X body is actually a chromosome. Okay. So, inside the sperm, they saw one extra body, and if that extra body is there, then that is a chromosome. And because the scientist has given the name as an X body, so the other scientists, those who have found that it is a chromosome, they have given it a name as X chromosome. Okay. Now, if the sperms has X chromosome, then that X chromosome is responsible for the girl child. And if the X, like if the sperms does not have anything, then that will be for the male child. So this was the method of sex determination that was given by the Henkin. Now, after all these discoveries and everything, at least we got one conclusion that there are two types of chromosome. Okay. Two types of chromosome. One is called as autosome and next is called as sex chromosome. Clear? Now this sex chromosome is responsible for the sex determination of offsprings. Okay. So this is about the sex determination. Now it is not compulsory every time that males should have heterochromosomes, okay, and females must have the homo, that is X and X should have must be there with the female, X and Y must be there with the male. It is not compulsory in all the animals. It is possible in case of human beings that males have the two chromosomes, that is X and Y as in sex chromosome, and females have the X and X chromosome. But in other organisms, there are some different things. Now see, what are those? Chromosomes. Now see, sex determination in case of some animals. For example, in case of fruit fly and human being, normal situation. Now what is the normal condition? Males have two type of chromosomes. One is X and another is Y. And females are homogametic, that is they contain X and X. This we all know because we have already studied this thing in our previous class also. Okay, that is females are homogametic and males are heterogametic. Now see what happens in case of birds. In case of birds, the males are homogametic. Okay, and females are hetero. Here we can write down as XY and XX also, no problem. But it may create confusion because we all know that X and X is the homogametic for human beings. Okay, for fruit flies. And that's why if we will write down X and X, then we will get confused that whether it is the chromosome of a bird male or female human being. That's why we will denote it with some different letter, and the letter is Z and Z. So if the birds are male, then they will be homogamete and we will represent them with Z and Z. And if heterogametic condition, then it will be Z and W. Okay. Now see next. In case of plant, how to determine the sex? If Y chromosome is present, then that will be the male. And if Y is absent, then that will be the female. So this is the condition in case of a plant. In case of grasshoppers, see children, there is only one chromosome and that is X. If the two X are present, then the grasshoppers or the roundworms will be female. Okay. And if only one X is present, now nothing is 0, like nothing is O, it is 0. If two X are present, then that will be the female. And if only X is present, then the animal is considered as a male. Understood? So these are the methods to determine the sex in case of these animals. Now, along with this, we have one more thing, that is sex determination in case of honeybees. Now see children, we all know that in case of honeybees, there are the drones and the queens. Now drones can form parthenogenetically. This we have already studied. That is, by the process of parthenogenesis, the drones are formed. So if the drones are formed, like in case of honeybees, if the drones are there, that is the male bees are there, then they are produced by parthenogenetically. And if they are produced by parthenogenetics, then they all will be haploid. And if they all will be haploid, then they will contain only 16 chromosomes. And the queen bees, they all will have 32 chromosomes. Okay. So, in case of honeybees, we have a different system to determine the sex, and that is called as haplodiploid sex determination system. Haplodiploid sex determination system. Okay, haplodiploid. Haploid, that is for the drones, and diploid for the females, queen bees, and by this method we can determine their sex. So this is all about crossing over and sex determination. Now we have to see some sex-linked traits and sex-linked inheritance. Okay. So that we will do in the next class. Okay. Thank you children.